Great. So now, for our finale, we'd like to speak with Dr. Michael Higley. Michael is a professor at the Kavli Institute for Neurosciences at Yale. And Mike's a great example of our annual grant cycle. So he received a grant a year ago for what's called cortical dynamics, which he'll discuss in a little more detail. And then we are really proud to announce that actually our SAP was so impressed with his research that we've decided to fund him for an additional year. And he'll talk about what he's going to try to accomplish over the next year. Mike, are you with us?
Yeah, so look, I'd first just like to say thank you so much for having this evening and giving me a chance to tell you guys, sort of what we've been up to, and at least get to communicate a little bit of our excitement and what we're up to the families that have just been so incredibly generous to support us in this work. So what we are primarily engaged in is trying to understand or phenotype, as it were, the kinds of brain activities that are associated with dysfunction of this gene RI1 in mice. And so, as Chris says, my name is Mike Higley. I'm an MD PhD in the Department of Neuroscience at Yale University. And this work is done in close collaboration with Jess Cardin's lab, also in the Department of Neuroscience as well. And, one of the things that Chris asked us to do is sort of let you know how we came to this project and really got involved with the community. And quite honestly, the way it worked for us is that we're a lab that primarily has worked in understanding basic brain function using mouse models, and an opportunity to sort of apply some of our work to a number of models of neurodevelopmental disorders came along. And we also saw then this opportunity from the SMS Research Foundation, and we applied with some of our best ideas, and we're incredibly fortunate and thankful to be funded. And it really started us on this journey, which is turning out to be incredibly interesting for us, and I hope valuable to the community and the research efforts directed at RAI1 and understanding some of the mechanisms underlying Smith-Magenis syndrome. So just to give you a little, and so again, so this is a project that was generously funded by the Research Foundation, and so as you've heard, quite a bit, Smith-Magenis syndrome ultimately derives from dysfunction or loss of function of the gene RAI1. And there's a lot of efforts, both supported by the SMS Research Foundation, supported by NIH efforts, by other foundational efforts as well, to look at cell signaling, how individual cells, especially brain cells called neurons, may work or not work, how the synaptic connections, the chemical connections between neurons might be disrupted, how that affects the activity of those cells, how the wiring, essentially the circuit map of brains, might be altered when RAI1 is not doing what it's supposed to do, and also how sort of the whole circuit works, how cells interact together. And what we think is that behavior is ultimately the product of these complex interactions of different cells and cell networks. And in fact, that's where our work has really come in. So there's a lot of amazing work, some of which you've heard from over just the last few minutes, at more cellular genetic levels of analysis. And what we've been trying to tackle is a little bit from the other end, trying to understand how gene dysfunction leads to altered brain activity and how that might contribute to some of the behavioral changes that we see in SMS. So our model is to work with mice. It's fairly common. And one of the things that I'm incredibly thankful for to work with this community in particular is it is a completely reasonable question to ask why we use mice to model such a complex disorder that is clearly affecting human patients, human kids. And it's a good question. And the truth is that there are many experimental methods that we can use in mouse models that are not accessible in humans. And so we would consider this what's called a preclinical model, which was we are using the mice to understand both basic brain mechanisms and processes that go wrong when RAI1 is disrupted, but also, and I'll touch on this very briefly in a moment as well, to use it as a test bed for understanding or for potentially vetting new therapeutic options. The idea being, if we see something wrong in a mouse and we develop treatments that sort of correct that in the mice, there's at least some reason to be optimistic that those methods might ultimately provide some benefit to the human patients as well. And so we use mice in which one of the copies of RAI1 is disrupted, is missing. And we use viral vectors, in one case, something called an adeno-associated virus, to express a green fluorescent indicator in the brains of these mice. So this is a protein that isn't there normally. We put it in with a virus, and when their brain cells are active, this protein lights up. And we could use fancy microscopes to use that lighting up to look at how brain activity is unfolding in sort of the awake behaving mouse, and also compare and contrast what we call wild type mice with mice that may be missing a copy of RAI1. And so we have recently developed a set of microscopic approaches, one of which is called, we call it mesoscopic imaging. And this is sort of a fancy word to mean sort of middle. So we are not looking at single cells, which we might refer to as microscopic. And we're not looking at, say, the whole brain as you might with functional magnetic resonance or fMRI, but we're looking at sort of a big picture of one big circuit, the neocortex, which in humans is the big part on top. And so we can look at the entire neocortex in an awake behaving mouse at one time and assess the dynamics of its activity. And I'm going to show you just a little bit of a movie that's just kind of fun to see what some of this looks like. And so hopefully you can all see this play. And so this is essentially a top-down view of a mouse brain. This is the front up, the back, and then in terms of the left and the right, you can actually see some of the blood vessels, these lines going across. And these flashing patterns of activity, I think I can play it again. Everywhere that it's sort of dark blue is quiet. Everywhere that it gets really bright yellow or red is an area that's active. And what I hope you can appreciate is that this is a mouse just sort of sitting there, hanging out, and the brain is incredibly active. There are patterns of activity. Some regions are active together, some are in opposition. These patterns change over time, depending on what the mouse is doing. This is essentially a real-time readout of how the brain is functioning and potentially how it drives behavior in the animal. And so we're using this approach really for two main goals, sort of for ongoing work. The first, as I said, is to understand how disruption of the gene RAI1 might lead to changes in the patterns of this kind of activity, essentially to come up with a neural signature of RAI1 dysfunction. And so I'm not going to present the details here tonight, but what I shared with the research panel just a few months ago was that we already have in our first year of funding some really cool results showing very interesting differences between the RAI1 mutant mice and their wild type siblings in many cases, leading us to think already that there are some signatures there that we can pull out, that we can study. And that leads us to the second goal and what is a big part of the second year of funding, which again, we're incredibly thankful to have received. And that's to also start looking at whether or not some of the therapeutic interventions might serve to correct or at least partially ameliorate some of these functional changes that we see. And actually one thing that I didn't mention as well is to also try to link those patterns to specific behavioral outputs in the mouse. And so again, these are our two major goals for the coming year. And we're really excited to continue this work. And so I'll just finish by pointing out we just had our first sort of in-person event as our joint labs just a few weeks ago. We are incredibly grateful to your community, and we're really enthusiastic about trying to give back from the generosity that you've shown with hopefully some important new biological understanding. So thank you.
Oh, you're welcome. And thank you for all the work that you do. We're really excited to work with you for another year. And, we look forward to collaborating with you for many years. And hopefully you'll also be able to look for something like an NIH grant at some point. Do you think that would be a reasonable?
Yes. Yeah. Sorry, I should have mentioned that. Absolutely. But, so most of what and actually this is maybe a good point to make. So most of the work that our labs do is funded by the NIH. These are sort of large grants that support major multi-year research programs. Quite honestly, I mean, the funding that we receive from you all from the SMS Research Foundation is quite literally an amazing seed grant. This is just work that we would not have been able to start. We would not have done without the support that you all have given us, and we are well on our way. I mean, to be honest, I mean, with the data that we've already got, we could start to put competitive, applications together. I think, with the second year of funding, we're going to be an amazing space to continue to get NIH level funding. And we expect to continue this for a long time.
Yeah,
But thank you.
So motivating. So we thank you very much. And we look forward to working with you. So have a great evening. And thanks again, Mike.
Thank you.